Hello, we are Group 1, and we will be trying to save coral reefs from temperature-induced bleaching.
Hey, Professor Alice. This is Samantha Bustamante, or Prophet. My main position on this project was keeping track of the intellectual accounting journal. I'm going to go ahead and present the problem part as represented per our poster. Just basically, coral bleaching occurs when the coral polyps become stressed with just a few degree temperature increase in the oceans in which they inhabit. And one of the main causes of this is actually believed to be climate change. And what happens is that the coral themselves can't handle the higher temperatures, so the dinoflagellates also become stressed once the coral becomes stressed, and they'll eventually be expelled. In the long term, corals will lose their source of food and eventually become diseased because they no longer receive the proper nutrients and eventually will die off, or also known as bleaching. And there are quite a bit of marine organisms that rely on coral reefs for food and shelter. They also become threatened when they can no longer live within the coral reefs. Therefore, the entire biodiversity of the ecosystem of that area will become a lot less. And this actually makes a lot of fisheries and even tourists who like to go scuba diving in these areas, they all become affected because the coral is no longer what it once used to be.
This is Eric Lee, and I will be discussing the goals for our project. Currently, there are a few solutions to temperature-induced coral bleaching. However, one thing that researchers have discovered is that some of the coral reefs in the Indian Ocean are actually adapting to the warmer waters. They discovered that the corals they found there were using symbiotic algae that were actually tolerant of the higher ocean temperatures, and as a result, the reefs continue to thrive in spite of warmer water. That being said, it's not as straightforward as simply introducing thermal-tolerant algal species into bleached coral reefs. This would require introducing a foreign species into a native environment, which could produce a number of complications. For example, the foreign species might become invasive and harm the reef ecosystem as a whole. Instead, we plan to create a solution to this issue with a genetic approach. Rather than introducing thermotolerant algae into the coral, our strategy is to find the genes that encode for heat shock proteins that give these thermotolerant algae their heat resistance. Once we do that, we can insert these genes into thermosensitive algae. With this gene, the algae will become less stressed due to changes in water temperature. The end result, hopefully, is that we allow symbiotic algal species that were previously sensitive to temperature changes to gain resistance. This, in effect, will prevent the algae from leaving the corals in the first place, which prevents coral bleaching. Using this method, our ultimate goal is to stop temperature-induced coral bleaching, which prevents further loss of biodiversity and helps maintain the health of our coral reef ecosystems.
Hi, my name is Marilyn Martinez, and I will be discussing the gene of interest portion of this project. After countless hours of researching possible traits, our team was able to narrow down and conclude that the best gene that we wanted to introduce was the CYME underscore CMJ101C. This particular gene can be located on the chromosome 10 of the cyanodiazoxion meroloi strain 10D. This gene has the ability to code for small heat shock proteins. It is part of the heat shock protein HISP20 family, which can be expressed in prokaryotic organisms as well as eukaryotic organisms. The purpose of heat shock protein is to act as protein chaperone, which assists organisms when it reaches a level of stress that may cause heat-induced protein denaturation. When the heat shock protein is activated, it begins to repair and refold the damaged proteins into their proper shape in order to carry out protein synthesis. With this gene present, we are hopeful that it may reduce coral bleaching.
Hello, my name is Stephen Pham, and I will be going over the methodology of our experiment. The first step was to find a suitable organism which contained a heat-resistant gene of which we can extract. We looked for various options, but ended up agreeing on using the small heat shock protein mentioned earlier. Once that was finished, we searched and found a suitable plasmid, but will be modified to suit our needs. The plasmid is the PMT NPT Gus plasmid, which will be covered in a moment, but the neomycin phosphorotransferase gene, or the NPT, and the Gus gene will be removed from the NOS promoter and replaced with our gene of interest. The gene of interest will be isolated from Cyanidioschizon malariae strain 10D with the use of CRISPR and Cas9, and will be attached to the NOS promoter of the agrobacterium of which the previous gene was removed. While still in the lab, our newly designed PMT CYME underscore CMJ101C plasmid is amplified and inserted into the zooxanthellae in hopes it acquires the heat shock protein. The ones that acquire the heat shock protein will be the survivors of being treated with antibiotics. The zooxanthellae is introduced to coral to observe interactions between them, the coral, and increased temperature changes within the water. If successful, it will be dutiful to obtain permissions to begin operations to save the coral reefs.
Hello, my name is John Mark Galloway, and I'll be talking about the plasmid design. The plasmid we as a team selected was used in the research collected by Michael R. Ten Lois and David J. Miller through the Department of Biochemistry and Molecular Biology at James Cook University of North Queensland, Australia. In cartoon A, the plasmid was the one that Lois and Miller used to amplify their gene, the NPT neomycin phototransferase gene, which was what was removed. We also removed the Gus gene. We kept the NOS promoter, however, from the agrobacterium to help amplify our gene of choice, which is a heat shock protein, the CYME CMJ101C, from the Cyanidioschizon malariae strain. From this, we hope to amplify the heat shock protein to be able to amplify that inside of the zooxanthellae, which was mentioned previously in the method section of our poster. Thank you.
My name is Mahajbeen Kausar, and I was a part of Team 1. We have tried to resolve the issue of coral bleaching due to high temperature rises, and I will be discussing some of the ethical considerations that have to be kept in mind. First off, we need to have experiments that are designed with transparency and accuracy, and in order to do that, we will have to record our experiment over a time lapse and repeat it several times to see if the outcomes are consistent. The next concern was the method of transformation. Basically, what we extract the gene from and what we put it into, the vector needs to be a safe form so that we are able to go ahead and introduce it to the ocean. We also wanted to extract the genes in the most safest of manners because of the fact that it might harm other organisms. Another concern of ours was the fact that there might be marine life that may react differently to the introduction of a new gene. So in order to see the changes in marine life, we have to monitor the effect on the coral as well as the surrounding habitat. And for that, other ethical concerns could be money, the funding that might be limiting us. And lastly, but most importantly, we think that we need to have a respect for the laws and regulations that have been set in order to prevent any harmful impact on the environment. So yes, these are some of the concerns that we as a team came up in doing our experiment.
Lastly, here is our DNA sequence and our references. Thank you very much, and do we have any questions?